This is another lesson in the series on finding the engram, and here we're going to be looking at one of the commonly used behavioral paradigms in the study of learning and memory, and that is fear conditioning. So in a fear conditioning paradigm, rats are trained to associate a tone with a shock. Rats will show a fear memory to the tone associated with the shock, but not a different tone not associated with the shock. So here we see the situation. A rat is in a cage. The cage has a metal floor here. The rat hears a tone. Now initially the rat's not afraid of the tone, and we can see that looking at blood pressure or the cessation of movement. Freezing is a common response when an animal's afraid. It doesn't really freeze much. It might orient to the tone. But now we play the tone, and then we give the rat a foot shock. Right, so here we see, of course, when it gets a shock, blood pressure goes up and it freezes. It's afraid. And if you repeatedly present the tone and shock and tone and shock, pretty soon, just the tone alone will produce a fear response, increased blood pressure, and the rat freezes. So this is a conditioned fear response. And there's another kind of fear conditioning paradigm used. It's called contextual fear conditioning. And in this paradigm, the rats are placed in a certain context, like a cage, cage A, and given a shock, or a series of shocks. Rats are then returned to their home cage, and if placed in the same training cage, rats will show a fear response. So you can just put them in the cage and not even shock them, but they will show the fear response. They will freeze for a period of time. Rats will show a fear memory to the context associated with the shock, but not a different context, like cage B, that was never associated with a shock. So if the rat got a shock in context A, it will fear context A. If it never got if it experiences context B but never got a shock in context B, it won't show the fear to context B. So the fear memories are specific to the appropriate context. Now, scientists have done a lot of neuroanatomy in this system here, and they have identified that the amygdala is a critical component in the system for conditioned fear learning to a tone, for example. The neural pathways are such that tone information goes into the amygdala, the shock information goes into the amygdala, and scientists think that LTP-like synaptic plasticity is happening in the amygdala, such that with repeated pairing of tone shock, the tone pathway alone can generate the fear response. And there's in another lesson, I go over some of that research, but it is well established now that the amygdala is a critical site in the learning and memory for this type of association. Now, by contrast, contextual fear conditioning seems heavily dependent upon the hippocampus. Now, we know from other lessons that the hippocampus does get information from all the perceptual regions of the brain, so the context and the shock are also converging in the hippocampus. And researchers have identified through experimentation, lesion studies and such, that the hippocampus is required for the animal then to show the contextual fear conditioning. So let's say we put the animal in context A, so cage A, and we do the training in this cage, so tone shock, tone shock. Now, if we just bring the animal back into this chamber, it'll show fear because that's where it underwent the training. But of course, it'll also show fear response to the tone as well. So it's really learning two different things. The chamber itself, it's showing a fear response to the chamber itself, but also specifically to the tone. And if we look then at these the contributions of these two brain regions, if you damage the hippocampus, the animal will not show fear to the training context, to the cage, but if you play the tone, it will still show fear then, right? So in other words, this suggests that the tone shock pairing is happening in the amygdala proper, whereas the fear to the context required an intact hippocampus. In the remainder of this lesson, we'll take a look at some current approaches to finding the engram using the fear conditioning paradigms. So the lateral amygdala is a brain region implicated in the tone shock condition fear memories. Let's imagine here we have a population of cells in the lateral amygdala, and it's these cells here that are making changes that are the engram for the tone shock association, the memory for that association. Now, using modern techniques, researchers can tag those neurons in the lateral amygdala that are activated during fear conditioning. So again, the idea is you've got the, let's say this is the rat brain, you've got the tone shock presentations. Changes are happening in the amygdala. Now we know the amygdala is lots of cells, so we have to imagine that there's an allocation process going on. Only a fraction of the cells are going to be the ones that will become the engram for that association, tone shock association. So what they were able to do using various techniques, they can tag any of the cells that are active during the training, tone shock, whatever cells that are currently active during the encoding process, right? So those cells then will be tagged. Now, later on, you're going to test the memory. So when the tone is presented later, the tagged neurons have an above average chance of being reactivated as measured by a different tagging system. So here were the original cells that were active during training, tone shock. Here is the test. You give the tone. The green cells became active. Now they tagged them with a different tag from the original cells here. And so then they could just compare the populations. If there's any overlap, and there was significant overlap, then those overlapping cells were active both during the training and during the test. So it's as if the overlap cells would represent the engram, the cells that get reactivated upon retrieval of the memory. So the significant overlap suggests that the originally tagged neurons become part of the engram, and their activity is a marker of memory retrieval. So by having two different kinds of tags, the scientists were able to determine that some of the very same cells that were originally activated during the training get reactivated during memory retrieval. So some of those cells are going to be allocated to the memory, and then they get reactivated when the animal retrieves that memory. They went on then to ask the question, could neuron allocation be experimentally manipulated? Could you somehow influence which cells in the population are going to become part of the memory? So using other techniques, researchers genetically engineered a virus to deliver DNA that increases CREB gene activity. We'll see what that gene does in a moment. This made some neurons in the lateral amygdala more excitable. So here, let's say the pink ones now are the ones that have been genetically modified to express higher levels of CREB activity. The question was, during fear conditioning, would the elevated CREB activity make the cells more likely to be allocated to the engram? Now, CREB is one of these really important genes that is implicated in the plasticity among neurons. You'll recall from the tag and capture model that recently activated synapses, like this one here, gets tagged. It's biochemically tagged in a way that then it's able to utilize the plasticity-related products, so it can capture them. This was the tag and capture model. And this accounted for how it is that LTP can be specific to recently activated synapses. Activated synapses are tagged, and then they're the only ones that can utilize these proteins and so make long-lasting changes in synapse communication. Well, it turns out that the CREB gene is one of these important genes. It's been characterized as a memory gene. Its protein regulates the expression of many of the plasticity-related products, including growth factors, structural proteins, signal transduction proteins, other transcription factors. CREB activity makes the neuron more excitable and so ready for synaptic plasticity. So you recall, late LTP seems to require protein synthesis, and one of the genes that seems to be important in that process is the CREB gene. So back to this experiment. Remember, they artificially, or they modified the CREB activity here. They increased the CREB activity in the pink cells. And then they trained the animals. So they were trying to make it so that the pink cells would be the ones that would encode the memory. During testing, the tone alone produced a fear response. So training proceeded, and the animal did have a fear memory. Active neurons, during the testing phase, the green ones here, active neurons were tagged, and that population was compared to the CREB manipulated population. So the neurons activated during the tone test were compared to the original cells that had their CREB activity elevated, and here were the results. The CREB cells were 3 times more likely to be reactivated during the expression of the fear memory. So evidently increasing a neuron's excitability made it more likely to be allocated to the engram. And again, the blue would just be the overlap, the cells that were reactivated upon the test, but were also activated during the training. It's as if then the overlapping cells here would be those cells that had been allocated to the memory. And as it turns out, because you can increase Krebs activity and make it 3 times more likely that that particular cell gets involved in the memory, it seems like the Krebs activity is a very important factor in which cells get allocated to a memory. So other studies showed that Krebs-elevated neurons had higher levels of LTP at their synapses. So it's possible that the increased Krebs activity makes these cells more capable of the synaptic plasticity that would make those cells part of the engram for the memory. Now, these results suggest that under natural conditions, so we're not artificially elevating Krebs, under natural conditions, amygdala neurons that happen to be more excitable are more likely to be allocated to the engram. So this is really just a fundamental discovery. If you ask the question, well, how does a brain know which cells to store which memories? This kind of research is suggesting that, within the amygdala here, there's a population of cells, and some happen to be a little more active than others. Well, the cells that are a little more active will be the ones that are more likely to be allocated for the next memory, for the next learning event. And then down the road, there's another population of cells that have elevated activity of some important genes, for example. So under natural conditions, the cells that happen to have more excitable, more Krebs activity and therefore are more excitable might be the ones more likely to be allocated to the memory. Now let's consider another experiment. So here we have, say, the red cells have elevated Krebs activity due to a virus that inserts a gene. But then, using genetically engineered mice and a certain toxin, researchers can ablate, that is, destroy only those neurons that expressed elevated Krebs, those neurons preferentially allocated to the engram during training. So here we've got the excitable cells here, increased Krebs. You train the animal, so they're more likely to become part of the engram because they have elevated Krebs activity. But then prior to testing, you destroy those cells. And so the question was, what has happened to the memory? If you destroy the cells that have been allocated to the memory, does the animal still have the memory? And what do you know, when tested with the tone 2, 5, 12 days later, the fear memory was gone. Ablating the allocated engram neurons erased the fear memory. Importantly, in control conditions, ablating non-allocated neurons had no effect on the expression of fear memory. This shows that specific populations of neurons are the engrams for specific memories. So if you destroy the cells, the pink cells that were allocated to the memory during training, the animal is happy, won't show fear to the tone. But if you destroy a different population of cells, here, these green ones here, and leave the allocated neurons, then the animal is going to be afraid of the tone. So it's not just destroying cells that will interfere with memory. You have to destroy those engram cells that are the memory in order to erase the memory. Now, after ablating the originally allocated lateral amygdala neurons, that here was those original pink cells that had increased Krebs, and then we destroyed them. So the sad faces there are the dead cells there. The animal can relearn the association of tone and shock. This shows that other lateral amygdala neurons, now these new red ones here, can support new learning. In addition, ablation of Krebs cells did not block a memory acquired before the procedure, again suggesting a different population of cells stored the engram for that memory. So the green cells might have been the engram for some prior learning event, and those animals still had that memory. They just didn't have the memory that was encoded by the cells that were destroyed. But destroying those cells did not prevent the animal from learning the tone shock association, because other cells in the population could do the necessary changes to make a new engram for that memory.